Okay, stop everything for a second. If you're following liquid biopsy development, well, the way we analyze blood might be fundamentally changing.
Hmm.
There's this paper, Science 2025, with a finding that's uh, honestly really counterintuitive. It turns out platelets, those tiny blood cells for clotting.
Yeah, the nucleate ones. No nucleus.
Exactly. We thought they were just like inert background noise, but this paper shows they're acting like biological sponges,
Sponges, yeah. Soaking up and, crucially, protecting cell-free DNA.
including the really valuable stuff, tumor fragments, fetal DNA, the very things current tests are looking for in plasma. But we've been throwing the platelets away.
It's wild when you think about it. We all focus on cfDNA, the cell-free DNA floating in the plasma, right? That's the cornerstone for non-invasive tests, cancer screening, prenatal checks.
Right, standard practice.
But platelets, even though they're the second most common cell type in blood, were just dismissed. No nucleus, assumed no relevant DNA.
So the protocols were designed to get rid of them, to get clean plasma.
Precisely. And this work suggests that in cleaning the plasma, we've been discarding maybe half the signal, or even more in some cases.
Okay, so first step for the researchers then was just basic confirmation. Do these platelets actually have DNA in them?
Yeah, they used high-res imaging, DNA-specific stains, things like flow cytometry.
Yeah.
And yes, they found about 8% of circulating platelets stained strongly positive for double-stranded DNA. So it's definitely there.
8%. Okay, not trivial, but does it do anything?
Well, that's the key physiological part. They looked at situations with low platelet counts.
Like in mice where they depleted platelets or human patients with certain conditions.
Exactly, like ITP immune thrombocytopenia, where platelet counts are severely low. And what they saw was a really significant acute jump in the amount of cfDNA floating in the plasma.
Whoa, so the platelets are normally cleaning it up.
That's what it looks like. A role in cfDNA homeostasis nobody really appreciated. They're actively clearing it.
And they nailed this down in vivo too, right, with the pregnancy study.
This is clever. They looked for Y chromosome DNA, the SRY gene, in pregnant women carrying male fetuses.
Yeah.
They found those male DNA fragments in the mother's platelets, but crucially, not in her other blood cells, her leukocytes.
So the platelets specifically picked up the fetal DNA circulating in the mom's blood. Wow.
Yeah. Which brings us to a really important question.
Okay, let me guess. If platelets are grabbing DNA, are they just grabbing random junk, genomic debris?
Or are they concentrating the important stuff, the rare fragments like tumor DNA that we actually want to find for diagnostics?
That's the million-dollar question for liquid biopsy, isn't it?
Pretty much. And that's what they looked at next, moving into the clinical potential.
Right, comparing the DNA found in platelets, they call it pDNA, versus the standard plasma cfDNA.
They did. In cancer patients, even pre-cancer patients, they look for specific tumor mutations, things like KRAS G12D or BRAF V600E, common drivers.
Yeah, could they find them in the platelet DNA?
Oh, yeah, readily detected. But here's the kicker. In their mouse models for colorectal cancer, the mutant KRAS gene, the cancer signal, was actually found in higher abundance in the platelet DNA compared to the plasma cfDNA.
Higher abundance, how much higher?
In 65% of the samples. So a majority of the time, the platelets had a more concentrated signal.
65%.
Yeah.
Wow, that's significant.
It really is. And critically, this wasn't just in advanced cancers. They detected ctDNA, the circulating tumor DNA in the pDNA from patients with really early stage stuff.
Like precancerous polyps, where plasma cfDNA is often just too dilute to find anything.
Exactly that. Those low tumor burden cases where detection is super challenging.
So the platelets aren't just passively collecting it. They're acting like a protected, concentrated reservoir. How does that work? How are they protected?
Well, the thinking, based on some secondary mechanism work they showed figure 2 in the paper, is that platelets internalize it. They can take up little packages called extracellular vesicles loaded with DNA, or even just naked DNA fragments directly.
And once it's inside the platelet?
It's shielded from enzymes in the blood nucleuses that would normally chew it up. It's protected.
Okay, so the big picture implication here is we need to change how we do liquid biopsies.
That's the argument. The innovation is realizing platelets are DNA sinks, and the implication is a new strategy: dual compartment sampling.
Meaning analyze both the platelet DNA, the pDNA, and the plasma cfDNA together.
Exactly, combining the signals. The hope is that this could seriously boost sensitivity.
For things like catching cancer much earlier. Or tracking minimal residual disease after treatment.
Yes, or even improving non-invasive prenatal tests, especially when the fetal DNA fraction in plasma is really low. It could make a big difference in those borderline cases.
All right, let's try and boil this down. If you take away 3 things from this deep dive, what should they be?
Okay, first, platelets, even without a nucleus, actively grab and store host DNA from the blood. That's a new physiological function we didn't know about.
Got it. Platelets equals DNA sponges. Second.
Second, this DNA inside platelets, the pDNA, isn't just junk. It includes clinically vital stuff like tumor DNA and fetal DNA.
Right, the important signals are in there. And third,
Third, analyzing this platelet DNA alongside the usual plasma CF DNA looks like a really promising way to make liquid biopsies much more sensitive, especially for finding disease early.
makes sense. Dual sampling for better results. Is there a catch? A limitation?
Well, the main technical hurdle right now is standardization. You absolutely need methods to isolate the platelets very cleanly without getting contamination from other DNA-containing cells like white blood cells. That needs to be rigorously worked out for clinical use.
Right, process matters. Okay, one final thought to leave folks with. If platelets clearing DNA helps control inflammation, what happens later?
Yeah, it makes you wonder, what are the consequences when activated platelets eventually release this DNA cargo they've concentrated? Could they be acting maybe as targeted delivery vectors for genetic material somewhere else in the body?
Huh. Targeted DNA delivery by platelets. That's definitely something to mull over. Wild stuff.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.